All right, let's talk exams. You ever look at a big 21-mark biology question and just wonder, what do they actually want from me? Well, today we are going to do a forensic breakdown of one of those questions. We're going to use the official AQA mark scheme to see exactly what an examiner is looking for, word for word, and how you can secure every single one of those marks.
Okay, here's our game plan. We're going to break this thing down piece by piece. We'll start with the biology of exercise and performance, then we'll dive into the incredible science behind making monoclonal antibodies. After that, we'll analyze the steroid test itself and then deconstruct the results. And finally, we'll wrap it all up with some key takeaways for your exams.
So, first things first, we're kicking off with the nitty-gritty of what actually happens inside your body during really vigorous exercise. Then, we'll connect that to how anabolic steroids can give an athlete an edge, which really sets the stage for the rest of the question.
Okay, let's get into it. Question 4.1. This is a super common type of question, worth 4 marks. It's asking for 2 effects of anaerobic respiration. But, and this is the important bit, look at that command word, explain. That is a massive clue from the examiner. It means you can't just list something. You have to say why it happens to get those points. And this right here is the key to unlocking those 4 marks. The points are given out in pairs. You can't just say muscle fatigue and expect a mark. Nope, you've got to link it to its cause. So you'd say muscle fatigue happens because of lactic acid buildup. Boom, that's a pair, that's 2 marks. Or you could go with heavy breathing happens to get the oxygen needed to break down that lactic acid. There's your other 2 marks.
All right, brace yourselves, because here comes the big one, a 6-marker. Now, these design and investigation questions can look scary, I know, but honestly, all they're asking is for you to put on your scientist hat for a minute. Let's see what a perfect answer actually looks like, according to the examiners.
So, a top-tier answer really has 3 key ingredients. First, your basic method, get a group of athletes, make them do at least 2 different exercises. Second, what data are you collecting? Well, you're going to measure their heart rate before and after, and then work out the average increase. But the third part, and this is where people lose marks, the control variables. You have to mention that you'd keep things like the athlete's age and the duration of the exercise the same to make it a fair test. That's crucial.
Okay, so the question takes a bit of a turn now. We're shifting from general exercise into the world of performance-enhancing drugs. It's another explain question, this time for 2 marks. So you know the drill, we're looking for that clear cause and effect relationship.
And there it is, plain and simple. The first mark is for saying the athlete gets faster or stronger. The second mark is for explaining why. And the reason is that steroids build muscle mass, which means you get more powerful muscle contractions. See? Point, then reason. Easy 2 marks.
Okay, so we've covered why someone might take steroids. Now the question gets even more interesting. We're diving into the sophisticated science used to actually detect them. And that brings us to the star of the show, monoclonal antibodies.
Okay, time for a quick-fire round. Just 1 mark for this one. It's a straight-up memory test. What do you call that special type of cell you get when you fuse a mouse lymphocyte with a tumor cell?
And the magic word is hybridoma, a hybridoma cell. This thing is like a biological superhero. It gets its power to make antibodies from the lymphocyte, and it gets its power of immortality, dividing forever, from the tumor cell. Pretty clever, right?
All right, so our hybridoma cell is ready to go. Now what? This next question, for 3 marks, wants to know the next steps in the production process. And check out the command word, describe. That means they want a step-by-step account of what happens next. So the mark scheme actually lists 4 main stages here, and you just need to hit any 3 of them to get full marks. It's pretty logical. Step 1, you clone the cell. This just means you make tons and tons of copies. So now you have many identical cells which are all churning out the exact same antibody. And finally, you just have to collect all those antibodies and purify them for use.
Now this next point is absolutely critical to understanding how these tests work. What makes the monoclonal antibody so special? The mark scheme couldn't be clearer. It is all about specificity. A monoclonal antibody will only bind to 1 single target. It's like a key that only fits 1 lock. In this case, that lock is the anabolic steroid, and the antibody will ignore everything else floating around in the urine. Okay, theory time is over. Now we get to put all that knowledge into practice. We're going to look at the actual test strip they used to screen athletes and figure out how to read the results. All right, let's get a good look at this test strip from the exam paper.
You can see it has a few different zones. There's the bit where the sample goes, then you have the test area, and right at the top, the control area. Understanding what each of these does is the key to the next few marks.
And here's a question you'll see time and time again with these kinds of diagnostic tests. What's the point of that control area? Why is it even there?
It's actually super straightforward. The control line is just there to prove that the test strip itself isn't faulty. It's a quality check. If that line doesn't appear, you just have to throw the whole test away and start again, because you can't trust the result. It's the test's way of saying, yep, I'm working properly.
And here we are, the home stretch. This is where it all comes together. We've got the results from 4 different athletes, and our job is to play detective. Who's positive, who's negative, and whose test is just plain broken? Okay, take a look at the image showing the results for athletes A, B, C, and D. You need to look really carefully at where the blue lines have or haven't appeared in both the control and test areas for each athlete. Let's zoom in on athlete B. You'll notice, well, there's nothing there. It's totally blank. So the question wants 2 things. First, what's the evidence that the test failed? And second, can you suggest a reason why it might have failed? This is a great example of a 2-part answer. For your first mark, the evidence is simply that there's no blue dye in the control area. That's it. For the second mark, you need a reason. The mark scheme gives you a couple of options. Maybe the free monoclonal antibodies didn't bind to the control area. Or maybe, and this is probably the simpler answer, the urine just didn't move up the test strip properly. Okay, last mark on the table. Looking at those 4 strips, which athlete tested positive for steroids? Remember, a positive result means a line shows up in both the control area and the test area. So looking at the image, yep, the mark scheme confirms it's athlete D. Phew, okay, we made it through all 21 marks. So, after all that, what are the big lessons here? Let's take a step back and think like an examiner for a second. What are the key strategies we can take away from this?
Honestly, if there's one thing to take away from all this, it's that just knowing the biology isn't enough. You have to know how to answer the question. That means really paying attention to those command words like explain versus describe. It means spotting where you need to give a point and a reason. It's about using the right specific terminology, understanding why controls are so important, and being able to read a diagram or a set of results without panicking.
Once you get the hang of breaking down questions like this, you're not just learning biology anymore, you're learning how to win at exams. I really hope that deep dive was useful. Thanks for sticking with me. Now, what's another complex topic you'd like to see us deconstruct? Let us know.